Okay, it's really great to be here, and thank you for the really kind introduction, Jürgen. I'm going to obviously be talking mostly about what I started and established and spent so many years working on in Jürgen's lab and with his help, and then sort of move a little bit more into some of the new things we've been doing with the method, and at the end I'll talk about something really new that I hope you guys will find somewhat exciting. So I thought, because this is sort of it's a school, so we may as well sort of delve into a little bit some of the details, I guess, of the method that we established together, and aspects of it that I don't necessarily usually talk about, but I think might be useful knowledge for you guys. Okay, so but before I get into that, let me just give a little background. So as many of us are here, we are interested in human brain development and sort of what are the human specific features. And the way I come at this is from an evolutionary perspective. And if you just compare human brain with, for example, a mouse brain, there's a lot of really big differences, and we've already heard about some of the issues that raises with modeling neurological disease. But I'm also interested not only in this sort of just this comparison, which we often sort of continually make, but actually sort of the general variety that you see in mammalian brains. There's a lot of information out there that we could potentially get if we look a little bit more broadly. So for example, why is the bottlenose dolphin cortex so large, and what are the features about that we may have in common, and what's different? So we're sort of interested in the evolutionary perspective, and what are the common features and the differences between different mammalian brains. I'm not really going to get into all of the evolution stuff that we actually do in my lab, because like I say, I think it's probably more useful to talk more about the actual method. But just to say that one of the specific question that we're more interested in is brain size and neuron number, which are of course related. And the reason we're interested in that is because it is generally thought that probably brain size and neuron number is what separates humans from our closest relatives. So although all the primates have quite dense brains in terms of neuron number per cubic volume of brain matter, humans have this, in addition, this very large encephalization quotient, which also takes into account body size. So at the end of the day, we just end up with this really huge number of neurons. And so what we're interested in is how that neuron number is determined. And so in order to understand that, of course, we have to go back to development and neurogenesis. And so these are just sagittal sections of a human brain over time. And these are some drawings to sort of help you orient yourself. And I just want to get into the development, because it really relates to the actual method, since that's exactly what we're trying to model. So the brain of all mammals starts as this neural tube, which is an epithelium. And like all epithelia, it has a fluid-filled lumen surrounded by epithelial cells. And during development, that neural tube expands at varying rates along the anterior-posterior axis, with the most anterior portion expanding the most in humans, and that's, of course, what will then give rise to the cerebral cortex. And if you zoom in on the wall of the cerebral cortex here, I'm going to focus on cortex just because that is sort of the region that we're most interested in, particularly from an evolutionary perspective. But as we've heard, the cerebellum is also a very important region, but I won't get into it. So in the cortex, you have a cytoarchitecture that looks like this, where you've got these neuroepithelial cells initially, which are probably not drawn all that correctly here because they're really much more fat and shorter. And they undergo symmetric expanding divisions initially, and that really helps this tissue expand in the tangential or lateral dimension and really balloon outward. And then those cells switch from these symmetric expanding divisions to asymmetric neurogenic divisions. And that means that they generate one daughter cell that stays a neural stem cell, so they self-renew, and the other daughter cell is more differentiated. And that can be a neuron, so this would be direct neurogenesis, and the neuron then uses the basal process of this radial glial neural stem cell to migrate out and position itself in the cortical plate. The cortical plate will then become the gray matter of the brain. And then, of course, those neurons will mature and send out axons inward, and that will become the white matter of the brain. These neural stem cells, though, can also generate these intermediate populations, and this could be intermediate progenitors, outer radioglia. There's actually quite a lot of diversity there. But in general, these intermediate populations themselves go through a couple more rounds of division and thereby magnify the number of neurons that are generated. So you can imagine that in order to generate a larger brain, there's a couple of ways you could do that. You could increase the amounts of these symmetric expanding divisions before switching to asymmetric divisions, and either by simply increasing that or delaying the switch. Another possibility is that there's an increase in these cells, and that could be through their increased production or because they themselves can also undergo more proliferative divisions. And there's actually evidence for both, at least when you compare rodents and primates. So if you look at stage-matched rodent brains, what you find is that initially they look really quite similar, but then at a certain stage, the rodent cortex really stops expanding in that tangential dimension, and actually it starts thickening in this radial dimension, whereas the human brain is really, as you can see, is really ballooned out. So that suggests that at least between rodents and primates, somewhere between them, there must have been a change that would allow for more expansion of these cells, or like a delay from this symmetrically expanding divisions to asymmetric neurogenic divisions. But also, there's a lot of evidence for expansion of these cells. Again, when you do comparative neuroanatomy between rodents and primates, you find that while both have this ventricular zone, which contains the cell bodies of these neural stem cells, cortical plates containing the neurons, both of them have a subventricular zone which contains these cells, but in humans and other primates we have this very large, dense, so-called outer subventricular zone, and this is populated by these intermediate populations. So there's evidence that probably both an increase in these expansive self-renewing divisions or a delay in the switch, as well as an increase in these intermediate populations may have been important in primate brain expansion. But we know virtually nothing about the mechanism of that, and we know nothing about the human-specific features. So which of these processes are involved in human-specific brain expansion compared with, for example, chimpanzees and bonobos? So although I won't actually get into that evolution side really in this talk, it's just important to keep it in mind in terms of why we need a human-specific model. And so I got started on this in 2010 when I joined Jürgen's lab. And at the time, these were the important papers that I had at hand and that I started working from. And so there were these really key papers from Yoshiki Sasai's lab, and you'll see that he is a recurring figure. And there was also this work from Suchun Zheng when he was in Jamie Thompson's lab, where he established the first neural rosettes, although I don't think he actually called them that at that time. And then, of course, there was work from Pierre Vanderhagen's lab, and this was work from Rick Livesey's lab, also showing rosettes. But all of these were, so they involved like sort of different levels of three-dimensional culture. So in all of these cases, there's at least a starting stage of embryoid body 3D culture, but then they plate them down. And the sort of progression along these was that, especially with these ones, they waited later and later to plate them down. So that was kind of important. I thought, well, it looks like if you wait even longer, or maybe you never plate them down, maybe you'll end up with something quite interesting. So I actually, a while back, I tried to sort of write down all of the key studies that were really important for establishment of this technique, and I actually wrote them down here. And really, all that I'm going to show you, the method that we developed is, I think it's really important to keep in mind that it's obviously built upon a foundation of many other papers and many other studies where people have been experimenting with different conditions and everything. And so all I really did was sort of put together all these different studies and try to figure out what was the optimum conditions. And these 4 studies were the ones that were the most influential. I tried out lots of different medias and lots of different things, but actually at the end of the day, it's really the pink boxed things that are really the key steps. And one sort of totally new step, which was just at the end, but again, it's something that's also done in other fields. So basically, at the end of the day, what we found worked was to use the media formulations from these really early rosettes, actually, but then just not plate them down. And then we use these U-bottom plates described in Yoshiki's lab, because then you get a really nice EB. And then for the differentiation stage, we found that this kind of media formulation from Pierre van der Hagen's lab worked very nicely. And then I think the real key was the realization that Matrigel, used for intestinal organoids in Hans Klever's lab, showed such a strong effect. We tried that with our tissues and found a similarly strong effect on the budding and formation of these neural tube-like structures. And then sort of the last step was the introduction of some agitation. That's something that's done widely in tissue engineering. We just applied it now in this context, and then you end up with really large tissues. So now, of course, the field continues, and there have been, we wrote this in 2016, and even, I mean, since 2016, there's still, it's been 2 years, and there's been many, many more, so I don't think I could even list them all. But I think the credit for a lot of this foundational work, though, definitely does go to Yoshiki Sasai, and particularly, and I actually have his picture up here because I think he deserves a lot of credit. His lab really established the first, what I would call true neural organoids, and these were these optic cup organoids, because these are fully 3D, and they're fully self-organizing, and you generate a complete tissue in these structures. Now, they were still quite immature, and so further improvements have led to more mature retinal organoids. But I would say this was a really important paper. And of course, now I'll get into, there have been a lot of new methods and independently established methods, as well as methods that have built upon our work, and sort of with the focus on deriving specific different brain regions. But the thing that I think all of the methods have in common is that at the end of the day, really what we're trying to do is just model really early development, really early neural development. And so I always try to keep in mind what's actually happening in development. I'm a developmental biologist, so I think about what's happening in developments and how can we mimic that as close as possible. And I come at this, like I say, I come at this from a sort of more basic development perspective. I'm not a trained stem cell biologist. The first time I cultured stem cells was, I looked up a protocol online and we ordered some iPS cells and I tried it. And so, but I think that's kind of one of the reasons why we established this method, because rather than, I think it would be sort of naive to think that we understand so much about these developmental principles that we can recapitulate it completely from scratch. We don't know all of the signaling pathways. We don't know how all the cells interact with each other. So what we tried to do instead was to let the tissue build itself. So stem cells have this amazing capacity where if you take cells from the inner cell mass of a blastocyst, those are of course pluripotent stem cells, they will try to do what they're programmed to do in vivo. So in vivo, these cells will undergo a series of morphogenetic events called gastrulation, and they'll generate the 3 germ layers. And the more superficial layer is called the ectoderm, and the medial portion of that is the neuroectoderm, which will then fold up and close, and that will become the neural tube. And it had already been shown, as I mentioned, if you take these inner cell mass-derived pluripotent stem cells, which are embryonic stem cells, and you just culture them as aggregates, that's an embryoid body, and they will start spontaneously trying to make the different germ layers of an embryo. So we decided to try to take this approach and basically just sort of support the tissue, allow it to do what it's already programmed to do, with as minimal intervention as possible. And so if you just make embryonic bodies and you do nothing, you'll get all the different tissue types. You'll get intestine in there, you'll get kidney cells in there, you'll get everything. But what Su-Chun Zhang showed with his rosettes was that if you take those embryonic bodies and you plate them down in what he called neural induction media, but I think that's probably actually not a correct name for it, because it's not inducing anything. All it is a very minimal media. It's just DMEM with N2, which is a very minimal supplement, if you know anything about it. So there's no serum or anything. And so what that does is that basically all the other germ layer identities, the mesoderm and endoderm, essentially die out. They're not really supported by that media. So we're not actually inducing anything. So when we call it neural induction, it's probably not the correct name for it. But like I say, it's what Su-Chun Zhang had already named it back in 2001. It really should just be called, I don't know, everything else killing off media or something like that. I can't think of a catchy name for it. But anyway, and so then when we take these tissues, which now have this nice ectoderm around the surface, which spontaneously starts forming a polarized neural ectoderm, and now when we put those in a droplet of matrigel, there's this amazing reorganization of that neural ectoderm, where so at this stage, when they're just floating, the apical surface is on the outside, the basal surface is on the inside. So actually, if you think about it from a development perspective, that makes a lot of sense, because in development you have the neural plates, and the basal surface is down, and the apical is out. And then it folds and closes up, and now the basal is out. And so that's basically what we're forcing the tissue to do here by putting it in matrigel. We're forcing it to reorganize and form these closed neural tube-like buds. And then with agitation, each of these buds can expand and get really quite large. And I'll come to the agitation, because depending on how you do your agitation, you can get larger or smaller. And obviously no agitation, then you get smaller organoids. So it's actually quite simple, because like I say, we're not actually really directing anything. The initial description of this method, we initially used these spinning bioreactors, but to be perfectly honest, we don't use those anymore. I didn't even buy any when I started my lab, because we've essentially completely switched to these orbital shakers. This is the company that we use in my lab, and I think in Jürgen's lab as well. And I just wanted to put this up there because I think a lot of the issues with, sometimes there's been some reports in certain papers and things that have said that using an orbital shaker does not generate nice organoids. I think there's a lot of, basically it's not a simple task actually figuring out what speed to put the shaking at, because depending on the throw orbit diameter of the shaker, the size of the dishes, and actually I forgot to put this up here, but also the volume of media, you have to change the shaking speed because the organoids will experience a different centrifugal force. And so this is what we use in my lab, and we don't change it at all because it works perfect. Yeah?
Does the amount of organoids per tissue also influence this?
Yeah. We haven't calculated that, so we've actually tried to work with a mathematician to try to figure out the forces and everything that's happening on an orbit. And he wrote down a list of variables, and one of them was number of particles, as he calls them, organoids are being the particles. But I don't think that one, when we discussed with him, I don't think that one was as important as there are other variables that I don't have here because we simply can't really control for them. Size of the particles. Organoids change in size over time, so ideally we might have to change the shaking speed as they grow. But we obviously we don't do that because we have all different staged organoids on there. It's not realistic to do that kind of thing. But yeah, there's a lot of variables basically, and what we found is that this works. And I can say that I'm going to show you our newer method where we have a cortical plate. And we went through a couple of months after I started my lab where we weren't getting cortical plate anymore, and it was because of the shaking speed. So that is how key this is. The organoids looked fine in every other way, but they weren't making a cortical plate, and it turned out to be because of the speed. And so this is the rule in my lab is we do not change that. But actually, like I say, we want to try to figure out what the speed would be for different size dishes and different amounts of media, because it would be good to be able to change. Anyway, okay, so that's sort of more of the details of that aspect. So of course what we end up with are these very nice organoids with lots of different regions, retina. You can see here by this pigmented region, which is the retinal pigmented epithelium. We have various forebrain regions. This would be the dorsal cortical tissue, and this would be ventral forebrain region, ganglionic eminence. We have choroid plexus, which generates the cerebral spinal fluid, and even on very early organoids, we can even see midbrain, hindbrain type boundaries. Now these kind of boundary regions we found quite interesting, and so a PhD student in Jürgen's lab and I worked together to try to investigate that a little bit more. So although the overall shape of the organoid doesn't look like a brain, obviously, it doesn't have, body axes, dorsal, ventral, anterior, posterior, but it did seem like when we found these kind of boundaries that maybe there is some relative positioning of brain regions within the organoid. So maybe not on a whole macro scale kind of organization, but maybe on kind of a meso scale. So we decided to look at the forebrain, because we kind of had to focus at least on one region to start with. And the forebrain's great because it has a sort of a linear arrangement, although it doesn't look like that, of these tissues relative to one another. So you have the choroid plexus in here, and it transitions into the hem, which is an important organizing center. And then from the hem, you have the dorsal cortex, which is actually highly influenced by hem. And then down here, you have the ganglionic eminences. And at the boundary between the dorsal cortex and the ganglionic eminences, you have the pallial-subpallial border. So there is this kind of arrangement where you should have a choroid plexus next to a hem. You shouldn't have a choroid plexus next to the pallial-subpallial border. So we wanted to try to just see if we see that kind of organization. And so what Magda did was she looked for organoids where she could see a choroid plexus, here marked by TTR, and a cortex. Now we like to use TBR2 actually as a cortical marker, because we haven't found a single marker that is reliable enough other than TBR2. Even PAC6, as I will show you, stains many different brain regions. So TBR2 is a very good marker and also seeing it localized properly in the subventricular zone is a very clear marker of cortex, dorsal cortex. And so then when she found these two regions next to each other, we asked, well, is there a hem in between? Because there should be if it's the right spatial organization. And so she did an in situ for Wnt2b and found there is a very bright spot for Wnt2b there, suggesting that there may be a hem there. But actually, interestingly, she found this really bright band over here. And so we thought, well, I wonder if there's actually another cortex over there. And when she went a little bit further in serial sections, she did indeed find a cortical tissue over there as well. So we could kind of predict that that would be where a cortical tissue would be. And so that suggests that at least that arrangement we seem to have. And we could also find, again, using TBR2 as a dorsal cortical tissue and now GSX2 as a marker of the ganglionic eminence, we could find this kind of border between those two regions as well. I think it's quite interesting because actually we had described in our first paper this kind of organization, but we didn't really characterize it very well. But we could see these regions that were positive for NKX2.1, a marker of medial ganglionic eminence. And we could see dorsal cortical tissues over here. And when we found those two tissues near each other, if you stained for interneurons, we could actually see interneurons that seemed like they were migrating in the right way and forming sort of a migratory stream, which is quite surprising. So not only do these tissues end up next to each other in the right sort of mesoscale organization, but also they seem to be signaling to each other. Now, as I think this illustrates the sectioning angle, sectioning plane is an issue. We have no body axes to guide us. And so it would be a bit like if I took Sergiu's brain out and I took Jürgen's brain out and I didn't use any body axes and I just randomly cut them up into slices, they would look completely different. You'd probably think they weren't even, the same organ or at least the same stage or I don't know. But or something else.
It would be wonderful.
Yeah. So that's kind of what we're doing with our organoids. We have no body axes, we're just randomly sectioning these things, and that's, you know, that's difficult. So that's why it's so important to use markers to really define where you are. But another way of sort of dealing with that is just don't section them at all. Yeah?
So do most of the organoids have like all of the different regions, or do you have some that are like one?
I'll come to it, yes. I will definitely come to that, yeah. So the other idea is to just not section it and image the whole organoid. Now, there are issues with that. So first of all, I will show you an organoid that we collaborated with Kwang-Hoon Chung at MIT, who developed Clarity. And he did some clearing of an organoid for us and light sheet imaging. And this is just DAPI staining, so you can see the overall sort of shape and architecture of the organoid. And it will trace through the ventricles, so you can see very nicely how the ventricles are sort of interconnected. And there's this highly folded choroid plexus down here. It won't trace out that part. It'll just trace out the ventricles of the dorsal cortical tissues. But you can see how nicely interconnected these tissues are and how elongated those ventricular spaces are. So this is really nice for seeing the overall structure. The issue, of course, is you're limited in terms of the numbers of markers that you can actually stain for using this kind of approach. So I have to be honest, most of what we do is still sectioning, classic sectioning and staining, because that way we can do lots of different marker analysis, and we can do a lot of quality control on a set. So we take like half of our 10 slides, let's say, and we stain for a whole bunch of quality control markers, even just histology, just to see the structure of it. And then the next 5 we can then actually do experiments with. Whereas with a whole cleared organoid, you don't know, you'll waste all of your colors, all your antibodies on just figuring out if that's a dorsal cortical tissue, and then you won't be able to look at anything else. Gosh, my screen is awfully bright. I can't actually see it. Right. So in terms of actually modeling what we're interested in, which is neurogenesis, although there are all these different sort of brain regions, and you don't necessarily know what you're looking at until you do the marker analysis, when we do look at, once we have identified a cortical tissue, we find that it does have the right architecture, and it has, progenitors on the inside and neurons on the outside, just like in vivo. And those progenitors very nicely model radioglial progenitors in vivo. They have the right morphology. They also undergo something called interkinetic nuclear migration, where they move their nucleus along the process timed with the cell cycle. We see various intermediate populations. I've already mentioned TBR2 here, but we also see these displaced radioglia, which are displaced from the ventricular zone. And these appear to be outer or basal radioglia. And of course, neurons are generated, and when they're generated, they properly leave the ventricular zone and cover the surface. And actually, something that Magda and I showed as well was that they're also generated in the right sort of timed fashion, which makes a lot of sense because Pierre Vanderhagen had shown that in vitro, in 2D, you get the right timed generation of neurons. But it's very good to see here as well. So we see first these reelin-positive cells, these Cajaloricious cells, and then we start seeing deep layer neurons and then more superficial layer neurons, and then eventually astrocytes. And we quantified this here, and you can kind of see the progression from reelin to deep layer, superficial, and then astrocytes. We also did some kind of, at the time now compared to current technologies, or current experiments, I guess, really, we just did some calcium imaging, and this is just a dye, but we could see some spontaneous neural activity, and hopefully it will come up. There we go. There's a neuron. So this is just spontaneous activity, and we could also activate it or increase its frequency by putting on the neurotransmitter glutamates, and I don't show it, but we could also block it with tetrodotoxin. And then I'm not really going to go into it because I think there has been quite a bit of focus on neurological diseases, but we were able to model this. Oh, something happened to my writing there. That says control, not roll. Anyway, and so anyway, we were able to model a genetic form of microcephaly using this system. So coming back to the variability in different brain regions and such. So there are, between different organoids, between different batches, there's a lot of variability. And so one way we deal with that is we just sort of focus on the region that we care about. we do a whole bunch of quality control, and we look specifically for the region that we care about. For example, this really large cortical tissue, and we just look at that. But that's very time consuming, and it means you throw away a lot of organoids, and it gets a little bit demotivating. So, and this is true for all the different brain regions. So sometimes we'll have a batch of organoids that has a whole lot of these retinal pigmented regions, and other times we have none. And I think that's demonstrated a little bit better when we actually do some marker staining. So here are 2 organoids from 2 different batches. And FOXG1 is a marker of forebrain, and you can see that both of them have some forebrain tissue, but this one has it much more sort of nice architecture. But it also has a lot of other stuff. And this one only has a couple of little rosette-like regions. And so when we do some more staining, we see that these are PAX6 positives. These are probably cortical tissues, but there's this really bright tissue here that's PAX6 positive. That's my guess would be that it might be hindbrain or spinal cord even, because PAX6 is actually very high in the spinal cord. OTX2 in this case, at this late stage, is marking more midbrain identities. And you can actually see, it's quite nice, you can see the sort of transition from these PAX6 positive, OTX2 positive, to OTX single positive, and then PAX6 positive alone. So it's almost like you're getting hindbrain, midbrain, forebrain, which is kind of cool. But I think it's just we happen to catch that in that section of Yale. So anyway, you can see there's a whole lot of different, and actually down here there's a region of probably choroid plexus. So there's a whole bunch of different identities, and you don't always get the region that you want. Now this one is okay, you could just focus on the region you care about, but this one is not really usable. So the way that many, this has been a common issue, obviously. So many others have addressed this by putting on growth factors or small molecules to basically direct differentiation towards a nice, clean neural identity. You get very clean tissue that way. And I'm sure that Sergi will talk more about his method, so I don't really need to go into details. But just to emphasize basically that I think the reason that we have all this variability is because we don't have any growth factors or small molecules in there. It's completely undirected. And others have actually taken the cerebral organoid method and then applied small molecules to make specific individual regions. This was work done in Guo Liming and Hangjun Sung's labs. And also in Jürgen's lab recently, they've shown that you can generate specifically dorsal or specifically ventral forebrain regions and even fuse them very nicely. And this was a series of 3 papers that all showed this really beautiful effect of if you take dorsal and you take ventral regions, you get a communication between the two, and you get interneurons properly migrating from the ventral into the dorsal. But again, something I'm sure that Sergi will talk more about. So, but in sort of looking at this approach, what occurred to me at least was that there's sort of this balance between homogeneity and complexity. So as you start adding more, I've just called them growth factors, but small molecules anyway, however you're affecting the signaling pathways, as you add more and more, you can imagine you get more and more pure. And so the purest in vitro system would just be clean neural stem cells. But those cells, of course, we probably wouldn't really want to use for many of our studies because they lack even the basic architecture of being epithelial, for example. And rosettes do have that epithelial characteristic, and they therefore also better model the lineage. So these guys, for example, don't make intermediate progenitors, but rosettes do. So there's something important about that tells me there's something important about the structure. It's this sort of classic structure-function relationship. And so as you get more and more pure, you get a little bit less of the large, complex structure. So I'm not saying that one is better than the other, actually, because I think it really depends on the question, and I'll come back to that. Because if you wanted to do some kind of screen or look at, I don't know, tens or hundreds of patient cell lines, organoids would not be the way to go because they are too variable. So, but the kinds of processes we're interested in my lab in terms of, cortical size determination and those tissue morphogenetic events, we want that complexity. So we're sort of using it as we're using it for investigating developmental biology and just sort of trying to understand what's happening there. And for that, it's really great. And I think this kind of illustrates also that trade-off between complexity and homogeneity, where, I mean, they're not totally matched, but they're kind of similar sized scale bars at the end of the day. Anyway, and you can see this difference in size. But it's not just size of the whole organoid, because if you just look at specifically the cortical regions and then sort of just measure those, now these are to scale, then you can see that the cortical regions are much bigger in the whole brain organoids. And as you get sort of purer and purer, they get smaller and smaller. So this basically led us to wonder if there would be a potential way to maintain the complexity, but sort of reliably. I don't know if that makes sense. Not directing its differentiation, because we didn't want to lose the complexity. But we did notice that sometimes we'd have a batch where all of the organoids were beautiful and made big cortical regions. So we thought, how do we do that reliably? How do we make every batch look like that? So we just tried to first investigate where this heterogeneity was coming from, and as we suspected in the first place, it does seem to be most of the heterogeneity comes from batch to batch. So if we made several batches here, and basically the take home here is that if you make organoids on, let's say, a Monday, and then you make some more organoids on a Friday, the ones all made on a Monday tend to look very similar to each other, but the ones made on the Monday, they also tend to look similar to each other, but they look different from the batch you made on the Monday or Friday or whatever I said. Anyway, and so the features that we're looking for, of course, is this clear neuroectoderm around the surface, and you can see these two batches look really quite nice. They have this nice neuroectoderm, whereas like this one is just really horrible, has these weird like frilly sort of rosette type tissues. This one you can't even really see a clear demarcation, a clearly demarcated border. So then we did some staining for different germ layers to try to figure out what's going on inside of these that's allowing them to organize so nicely, or what is their organization really. And we found that N-cadherin was on these good organoids, very nicely sort of coating the surface. You have this very nice polarized neuroepithelium around the surface. And you have sparse labeling for other identities, but they are there, which I'll come back to. Whereas in the sort of bad organoids, you have a lot more of those other identities, and the neuroectoderm is not well organized. It's sort of in patches. So this kind of got us thinking, well, the tissue that we want is covering the surface, and if we want more of that tissue, then we probably shouldn't start with a round, spherical EB. That's going to have the lowest surface area to volume. Let's try to come up with ways of increasing surface area to volume. And one way is to just decrease the size of embryoid bodies, and that actually does work, but of course then you end up with much less starting material. So what we decided to do was use these microfilaments, which are really just shaved off little pieces of dental sutures. So they're nothing really all that fancy. It's a good material because it's absorbed over time. So these are just absorbable sutures. And when you put in a couple of these small microfilaments, so they're about 1 millimeter in length and 15 microns in diameter, and then you put them with the embryonic stem cells in the U-bottom plate, then the stem cells will coat it. But one important feature is that we put in a lot more stem cells than so we don't put in enough fibers basically for the amount of stem cells. So what I'm saying is that if you calculate out sort of the size of the fibers relative to the size of the size of the cells and their number, what you find is that about 5 to 10% of the cells are touching the fiber, but the other 90 to 95% are not actually touching the fiber, which is an important feature because it's very different than typical sort of fibrous microscaffolds, where the goal there is actually that the cells are embedded in a scaffold, in a fibrous microscaffold. But in this case, actually, the fibrous scaffold is embedded in an embryoid body, really. So most of the cells are still just touching each other, and that means they're still self-sorting out and self-organizing. And so basically, we're not really changing anything about the embryoid body, just the shape of it by doing that, yeah?
I have a question then. What about the number of cells? Like, did you change the number?
Yes, yeah. So normally in the original protocol, we described 9,000 cells, and with this, we do 18,000. And that seems to work well. It was just empirically figured out, though. Okay.
Excuse me, and this is constant from line to line, the number of cells?
Yeah. Yeah, so it doesn't seem to, I mean, there are different, so we can come back to that. Different lines have different propensities to make nice organoids, and some lines you wouldn't necessarily need to do this, because some lines just make nice organoids even as around, because they seem to be sort of biased towards neural. But more difficult lines, this tends to work better.
So I guess to follow on the question, do you have to adapt this based on line? Because I think that's what
you have to adapt everything based on lines, really, at the end of the day. You have to adapt everything based on individual batches. So like every time we make organoids, we look for these morphological hallmarks, and we adapt the timing according to that. So it's a bit like, I don't know, gardening or something. It's like if your plant is running out of water, you add water. You don't just automatically water every day, right? I think it's like that, and that works best. If you just blindly use timing without really thinking, then it doesn't work as well. So the hallmarks, for example, like in this one, once you see this nice neuroectoderm with a boundary where you've got a really nicely demarcated, then it's ready to go in major gel. And so, yeah, so these are different features that we describe in our protocols paper. And this just basically makes it happen more reliably. So, right, and so then what was another interesting feature, so we found that they did generate more of the neural tissue more reliably, but what was kind of surprising as well, and we didn't expect this, was that they also seemed to be biased towards forebrain identity, which is quite interesting because we're not actually putting any small molecules on there to drive them towards that identity. And although we don't have an actual explanation for this because we haven't done any experiments on it, I have sort of a hand-wavy idea that it might have something to do with the abundance of those other non-neural identities. Because if you think, again, from a developmental perspective, if you think about the neural tube, the most anterior portion of the neural tube is a bit isolated from the rest of the embryo, from the mesendoderm, basically. And so by limiting the amount of those identities, it may not be receiving the signals that it would have received if there were a lot of mesendoderm that might posteriorize it. But we haven't actually tested it.
I mean, I think, right, I guess the correlation is biased because that's only the data.
Yeah, I'm only showing that here. We have in the paper we've done looking at neural ectoderm versus non-neural identities, and those are clearly down. They're clearly way down.
How about other brain regions? Because that was my question.
Other brain regions. Yeah, I mean, because this is from the Allen Brain Atlas, and they just don't really have, they don't have hindbrain on there. I mean, they have cerebellum. But
Hawks before or something else?
We did. So we looked at specifically, and so we did that. That's also in the paper. We looked specifically at Hox genes and things like GBX, which would mark more hindbrain identities, PAX2, and they were way down, yeah.
And this is consistent because I guess that comes back to the IPS lines, and I want to put this into context as we're moving forward with the other propensity of different lines to make different fates. And I think yesterday we had a conversation, one point about, I think with some of you, about the propensity of human embryonic stem cell lines to make different cell types, right? For instance, H6 versus H1 versus H1.
So we test,
H9. So
we tested 3 cell lines with this method, with and without the fiber, and all of them were improved. They were H9s, H1s, and a commercially available IPS cell line. Now, in my lab, because we're sort of getting more and more cell lines, what we find is that this does improve even difficult cell lines, but there are some cell lines that no matter what you do, you can't make organoids. I think they're not pluripotent because we can't make any neural tissue at all. But this does seem to help some cell lines that are more, let's say, finicky, where I think what it does is it's not like a magic bullet. But for example, if you have a cell line that has an efficiency of 10%, you make 10 batches of organoids and only 1 is a good batch, then this will bring that rate up. but depending on the batch and sort of what that starting point is. If it's 10%, it might bring it up to 40%, but if it already started as 40%, it might bring it up to 80%, so yeah.
Sorry, Madeline.
Yeah.
What is your interpretation for how the fibers suppress the alternative identities of the mesenteroderm?
I really think it's just the surface area to volume and the way the cells sort out. When they start differentiating, there's clearly this self-organizing patterning where the ectoderm is always on the outside of the embryoid body and the mesenteroderm is on the inside. And so for example, if you make a really big embryoid body, which is going to have actually a lot more of that central material, those tend not to make good brain organoids because you end up with a lot of other identities in there. So I think that's all it's doing. Yeah. Okay, and then this is just some marker staining again, where you can see very nicely the reproducible formation of forebrain. This region down here is actually choroid plexus and dorsal cortical lobules, kind of. So now this is an example of one where you can see really nice production of these very big, what we call cortical lobules, nice big ventricles, and quite reliably. So most of these tissues are cortical tissues. And we can, again, we can look specifically at these individual lobules, and we can see that, from organoid to organoid, various different stains, they always have the same really nice architecture. And so we can model neurogenesis quite nicely. Yeah?
Does the fiber even get like an anterior posterior type of signature? Does it change?
No, it doesn't. Yeah, no, that's, yeah. So the other question that we wanted to look at was the neuronal positioning. So it had been described, we described the sort of rudimentary separation of deep and superficial layer. Others had described this more extensive separation of layers. And I thought that was quite interesting because actually, if you look at early stages, basically I started wondering how it manages to do that. Because early on, those neurons in both, these are both images from these two papers, the neurons don't really seem to be well organized. And what I mean is, if you look in vivo, the cortical plate has this very clear organization of neurons being very nicely aligned and very dense. And it's been shown in vivo that that formation of that radially organized and dense cortical plate is a prerequisite for formation of the 6 layers of the cortex. So I started wondering how we could maybe recapitulate that. And one of the things that we thought about was, well, of course, in vivo you have this tissue covering the surface of the brain, which is the meninges, and that's obviously not generated by the brain tissue itself. And that is helping support and maintain the basement membrane that the radioglia are then attaching to. Of course, the radioglia are the guide for the neural stem cells, so you might imagine that if the radioglia are not properly attaching, that the neurons won't actually know where to go correctly. So we looked at whether the basement membrane was even present, and although we saw it early on, which makes sense because early on they've got the matrigel basically, but later, after they start making neurons, the basement membrane really seems to break down, and whatever is left of the basement membrane is kind of stuck in here next to the ventricular zone rather than on the surface. It should be, outside the neurons. So we tried a whole bunch of weird things, and it turned out that actually the simplest experiment worked, and I probably should have done this first. But basically, we found that if you just dissolve extracellular matrix components, and the easiest way to do that is just matrigel, just dissolving a small amount of matrigel in the media, that the organoids now, the way this is drawn is probably not very accurate. This is actually, so we put this in later after the organoids have either grown out of the matrigel, as they sometimes do, or sometimes we actually cut the matrigel off, or we sort of gently pipette them out of the matrigel. We let them make neurons for a little while, and then we dissolve the extracellular matrix components in the media. And that causes somehow the brain tissue, the brain organoid, absorbs the components from the media and maintains the basement membrane really nicely. And so, long story short, because that's published, is when we put that dissolved ECM in with our fiber method, then we can get this very nice cortical plate. And you can see it really nicely aligned. And so this is pretty cool because we can do some experiments with electroporation because we have these very nice large cortical lobes that we can actually inject into. You can put in a plasmid along with dye so we can see it, and then you basically electroporate it. Now again, because I'm coming from a development background, this is something we typically do in mouse development. It's called in utero electroporation, except here it's in organoid electroporation. And this is an example of an organoid where I've already injected the plasmid along with this blue dye. And so you can see very nicely this nice large lobe that I've injected. And then we can label with something like GFP, and we can do live imaging. So this is one of these movies where you can see this is actually an outer radioglia that's going to pop up and divide in just a second. There it goes. And you can see these radially migrating neurons. And this guy down here, my best guess is an interneuron, but without a marker, I can't say for sure. So it's pretty cool because we can specifically label neural stem cells at different time points. We can watch neurogenesis in action. We can watch neural migration. We have a cortical plate now, so we can look at neuronal migration defects. And we can even look at other events, which I will get to in a moment. And so this is just kind of an overview slide that I thought I would put up. Not everything is on here. There's been a lot of work. But I just kind of wanted to highlight some of the things. I told you about this kind of endogenous spatial organization, and I mentioned the microcephaly. We also did some transcriptomics and epigenomics to try to describe the molecular signature and found that it's quite similar to fetal cortex. Others have used it to look at things like Zika virus. This list is very short. It really goes on. And now, very recently, hopefully some of you have seen the transplantation study that was done in Rusty Gage's lab. And of course, these light-sensitive neurons that were detected in Paula Arlata's paper. And so I have this kind of future direction slide up here, just mentioning some of the things I hope that brain organoids will give us and where I think we need to go. Obviously, the main sort of use, I guess, that a lot of people are using them for is to model disease. But hopefully we can get to a point where we can do some drug screening. I did mention already the issues with reproducibility, and I think probably the more, it's one of those sort of trade-offs. And for example, in my lab, we're actually doing a study of certain compounds, and we're doing it in rosettes because we can't do it in organoids. It's just not realistic. But if we first do something in rosettes, then we can go back to the organoids. And so what I want to emphasize is there's a lot of different methods out there. Some have argued that we need some sort of centralized method that everybody uses. I actually don't think so. I think we should have lots of methods because each method has its strengths and limitations, and they can be supportive to each other. And it's kind of like when cell lines were first established. As we now know, having lots of different cell lines is useful. And if all of us just only used HeLAs, that would limit things quite a bit. So I think it's good to have several different methods. But again, I think you just have to think about what your specific question is and which method is right for that question, and not just choosing a method because it's sexy or it looks pretty or whatever, but actually, according to the question. And I'm hoping, we're not working on this, but hopefully somebody will figure out a way to there's been some vascularization with transplantation in vivo, but in vitro would be really cool. Patterning them, giving them axes, things like that would be very nice. And then I put this up here because although we want to maintain the complexity, if you think about the actual human brain, our brain is complex, but it does actually develop reproducibly from person to person. We all have the same, probably. I mean, I don't know, maybe if we did an MRI, we might find somebody who only has one hemisphere in this room. But generally, we all have the same overall architecture. And so if there's a way to do that, and maybe with patterning, that would be very powerful. So now I'm going to switch to a different topic, and I think we can turn off the video because I'm going to talk about something new that's not published.